In this video, we're going to look at a few brief study scenarios to determine if they show that the samples were independently selected or if they were selected in a paired fashion. Let's start with the first one. A new flea collar is tested on 23 randomly chosen calico cats, and the results are compared against 23 randomly chosen tabbies. So you want to ask yourself, we've got two samples here. Did the selection of the calico cats have any influence or dependency upon the selection of the tabby cats? The answer is no. So these are not paired samples. If we needed to compare these samples and assess the effectiveness of the flea collar, we would probably use a two-sample t-test. So that's the regular two-sample t-test, as long as the requirements for its use were met. And if they weren't, then we could look to the Wilcoxon-Mann-Whitney test as a backup. Let's look at the second example here. A new flea collar is tested against a eucalyptus bead flea collar. 15 labs wore the new collar for 30 days, and then they wore the eucalyptus collar for the next 30 days. Okay, so there's two sets of data again. We've got some sort of measurement of maybe flea counts for 30 days with the eucalyptus collar and the new collar. But were those samples paired? The answer is yes, those are paired samples. But why? Well, it's the same dog, right? It's like the same subject. Same dog is being exposed to two treatments. So we would probably use a paired test, like the paired t-test, to compare these samples. Let's look at the third one. Education and monthly salary are thought to be closely related in many fields, such as biology. Salary and years of education were correlated among 120 biologists working with the government jobs and 120 biologists working in the private sector. Two samples, paired or not paired? I'm going with not paired. I've got a couple more examples here I'll scroll down to. Let's start with the melatonin that was compared against conventional sleeping pills on a sample of 230 people with insomnia. For 2 weeks, the insomniacs tried the conventional sleeping pills, and then for another 2 weeks, the same people with insomnia tried melatonin. The total hours of sleeplessness was recorded for each person and compared. Okay, paired or not paired? I'm going with paired. It's kind of like the dogs example, right? There's the same individual, same subject, being exposed to the two treatments. Let's do the last one. 36 sets of twins were inoculated. We could probably stop right there, right? Because of what keyword? Twins. But let's continue on. 36 sets of twins were inoculated with a particular strain of flu, probably influenza, to test the effectiveness of the flu vaccine. One member of each set of twins received the vaccine, while the other did not. The genetic similarity of the twins provided a reliable mode of comparison for the recovery times between the vaccine recipients and the controls. Twin studies are so cool, right? They get to serve as their own control group with. So, what's the answer here? Paired or not paired? You got it. Hopefully that helps identify a paired design. And in the back half of this video, we're going to look at some notation that needs to be established for us to discuss formulas for the paired t-test. So, some notation. We're going to talk about differences a lot, so that's going to be represented with the letter D for difference. And that difference we did in the previous video. It's y1 minus y2. That's D for difference. But we often look at the mean of the difference. So this is D with a bar, mean of the differences. Kind of like way back when we did not just y, but y with a bar. This applies to the sampling distribution. So a little technical there, but thought I'd throw it in there. The parameter that we infer to, in the previous videos, we would say mu1, mu2. The mean population mean length for the rainbow trout, the population mean length for the cutthroat trout. It's a little different here because we're calculating differences, but we still want to infer to the population mean of those differences. So population mean, difference. The standard error of the mean difference is given by s over square root of n. Just like we saw s over square root of n a long time ago, but I'm going to be a little more specific here. I'm going to put little d's on these. So that's the standard deviation of the differences and the sample size of the differences. So I'm going to write those over here. S standard dev of the differences, and n is still sample size, but as it applies to the differences. So number of differences, you might even say number of pairs, number of differences, or pairs. It's the sample size. Now let's discuss the formula needed to perform the paired t-test. Before we do an example, it might be good to get that established. And so we need to discuss the test statistic. So let's look at the formula for the test statistic. Now this is called a t-test. So the test statistic is still called t. It's calculated from a sample, and it has the same structure as the test statistic from the regular two-sample t-test. And so this starts off with some sort of average. Remember with the regular two-sample t-test, it's sample mean 1 minus sample mean 2, and downstairs there's that big square root standard error. Technically there's a -0 there. So this test statistic for the paired test is going to look very similar, except it needs to be applied not to the individual sample means, but to the mean of the differences. So over here I'm going to actually write D with a bar, which means I need to define that over here. D with a bar is the sample mean of the differences. And technically there's also a -0. And downstairs we have the standard error. It's the s over square root of n as applied to the differences. And that's really all we need to discuss for some of the distinctions and notation with the paired t-test. So I'm actually going to cross this out over here. I don't want it to get jumbled up. In the next video, we'll do an example of the paired t-test.